Okay, so hello and welcome back. We're beginning a four-part series on carbohydrate physiology and its related metabolic pathways, with the first installment centered on fuel sources, storage, and metabolism in humans. Now, the first part covers an overview of nutrients that supply energy, how they are stored within the body, and the metabolic routes that transform them into usable energy. It also explores dietary carbohydrates, detailing their sources, digestion, and absorption, and additionally, it examines key glucose metabolic pathways such as glycolysis, gluconeogenesis, glycogen metabolism, among others. Now, carbohydrates, fats, and proteins are the three major macronutrients that supply energy and support vital function. Carbohydrates are the body preferred quick and fast fuel stored as glycogen and used especially during high intensity activity. Fats provides the most energy per gram and serve as long-term reserves, structural components, as well as hormone precursors. While proteins primarily build and repair tissues but can be used for energy when other sources are scarce. It follows distinct metabolic pathways to produce ADP, the body usable energy currency. Well, there are two main circulating fuel in human. The primary and most efficient form of energy storage in the body is triglycerides. While muscle holds the greatest amount of glycogen, larger than the liver glycogen, but it is the liver's glycogen that primarily supplies free glucose to the bloodstream, or the principal reservoir of glycogen from which free glucose can be released into the circulation. Now, when we examine fuel reserves in human, in a 70 kilogram man after an overnight fast, fat stores can amount to approximately 15,000 grams, supplying around 135,000 kilocalories of energy. Meanwhile, glycogen stored in the liver and muscles provide only about 2,000 kilocalories. This clearly demonstrates that fat serves as the body's largest and most efficient energy reserve. Now, illustrated here, the sequence of fuel utilization by the body to different physiologic state, from fed to fasting to starvation, highlighting carbohydrates as the primary energy source initially, fat as the main fuel during prolonged activity or fasting, and protein reserved for emergency energy use only. So why protein isn't the body preferred fuel source? Now, besides its role as essential biological component, protein is the least efficient energy source compared to glucose and fat. Additionally, the body doesn't have a dedicated storage system for protein. So it's typically used as an energy fuel only when glucose and fat are insufficient. Moreover, there are at least four reasons why protein or amino acids are preserved or aren't the first choice as fuel. Amino acids are needed for the building uh, and repairing tissues, enzymes, hormones, and immune molecules. Burning them for energy risks compromising this essential function. Number two, there is no protein storage or depot. Using amino acids for energy breaks down important protein, causing muscle loss during starvation or illness. As far as metabolic costs, converting amino acids into usable energy by via gluconeogenesis or the Krebs cycle is metabolically expensive and slower than using carbs or fat. And lastly, breaking down amino acid produces nitrogenous waste, which is ammonia, which is converted to urea and then detoxified and eliminated by the liver and kidneys. So, when does the body use amino acids for fuel? Number one is for starvation, or during starvation or prolonged fasting. Once glycogen is depleted and fat stores are tapped, the body may turn to muscle protein for gluconeogenesis. Number two, extreme endurance or cachexia in marathon runners or patients with chronic illness. Protein breakdown may contribute to energy supply. So, why carbohydrates are the body preferred fuel over protein or even fat? Now, that is because carbohydrates provide fast, easy to use energy and spare protein for other functions. So, as a summary, the key insight into the fuel utilization and storage: number one, glucose efficiently powers the brain, red blood cells, and active muscles with very little metabolic cost. Number two, protein sufficient carbohydrates intake protects muscles by reducing the need to use amino acids for energy production. Number three, using protein for energy produces ammonia, which the body must convert to urea, placing extra strain on liver as well as the kidneys. And number four, carbohydrates are quickly absorbed and used, making them the top choice for immediate energy during exercise and mental activities. Now, let's shift our focus on the organs that form the gastrointestinal tract, including the mouth, esophagus, stomach, small intestine, and the large intestine, or the so-called colon, rectum, as well as the anus. Now, this comes into direct contact with the food and digestive content. As you can see, all the different location, mouth, stomach, small intestine, there are enzymes for digestion, for carbohydrates, protein, and fat, okay, as well as absorption. Now, during high carbohydrate meal, the question is there any glucose or monosaccharides that is present in the diet? And the answer is practically nothing, because it contains polysaccharides and disaccharides like starch, amylose, amylopectin, etc., sucrose, lactose from the milk, and maltose. The digestion begins in the mouth with salivary amylase and continues in the small intestine with pancreatic amylase breaking carbohydrates down into maltose, isomaltose, sucrose, and lactose. In the small intestine, in the brass border, it contains alpha glucosidases, an enzyme that breaks disaccharide into monosaccharides before they are absorbed by enterocyte. This is the prerequisite. This is quite peculiar because in amino acids or peptides, dipeptides can be absorbed, not only single amino acid. Okay. So the sodium glucose cotransporter 1 facilitate the uptake of glucose into the enterocyte from where it is transported into the bloodstream. Now, after eating, blood sugar rises briefly as insulin helps cells absorb glucose. This rise is quick and controlled in healthy people but can be higher and last longer in diabetes, increasing health risks. The alpha glucosidase inhibitor, an agent that is available in the market, work by competitive blocking alpha glucosidase enzymes, and there are at least 3 different drugs in this class used to manage postprandial blood glucose level, as you can see there. Another potential agent is the sodium glucose cotransporter 2 inhibitor, which has some selectivity for SGLT1 and may reduce glucose absorption in the small intestine. Now, glucose, this is the mechanism of absorption of monosaccharides by epithelial cells of small intestine. The glucose is absorbed in the intestine by cotransporter with sodium via the SGLT1 transporter on the apical membrane that is on your left, the sodium moves down its gradient, helping glucose enter enterocyte against its gradient. Inside the cell or enterocyte, glucose exits through GLUT2, a glucose transporter, into the bloodstream on the left side. The sodium gradient is maintained by the sodium potassium ATPase pump, which uses energy on the basolateral membrane on your right side, which actively pumps sodium out to keep the cycle going. The glucose absorption by SGLT1 and SGLT2 in the kidney tubules operate in a manner very similar to that in the intestine. So not all SGLT2 inhibitor have the same selectivity when it comes to blocking SGLT1 versus SGLT2. Inhibiting SGLT1 can affect glucose absorption in the small intestine. As you can see, these are all more of an SGLT2 inhibitor that inhibit glucose reabsorption in the tubules in the kidney, giving rise to a lowering of blood glucose. Both sotagliflozin and canagliflozin have actively or have activity against the SGLT1 transporter, which could somehow help reduce blood glucose to the intestinal mechanism. So absorbed glucose level through the portal system to the liver, okay, after dietary absorption of glucose. Once there, glucose follows a multiple pathway. It can undergo glycolysis and enter the Krebs cycles to produce ATP. Or surplus or excess of glucose is converted into glycogen through glycogenesis for storage. No? If there is still excess, it can be transformed into fatty acids, which is known as de novo lipogenesis. Additionally, glucose can be released into the bloodstream to supply other tissues and organs for cellular uptake. Now we're gonna talk on de novo lipogenesis in the liver. And this happens when excess glucose is metabolized in the liver, where high ATP levels inhibit isocitrate dehydrogenase, causing citrate level to accumulate and be transported to the cytosol. There, citrate is converted into manonyl-CoA, the committed step in fatty acid synthesis. Now, manonyl-CoA also inhibit CPT1. CPT1 inhibition will block fatty acid entry into the mitochondria and ensuring that fat synthesis and breakdown don't happen simultaneously. Now let's go now to the glucose metabolism. Glucose metabolism includes various pathways, but glycolysis and gluconeogenesis are the most crucial. A solid understanding of these processes is essential for anyone studying diabetes. Now this illustration shows both pathways with glycolysis on one side and gluconeogenesis on the other. Glycolysis involves 10 enzymes, but only 3 of these steps are irreversible. Now, to understand gluconeogenesis, you just need to learn 3 additional enzymes that bypass these irreversible steps in glycolysis. During glycolysis, the conversion of glucose to pyruvate involves 3 key irreversible enzymes, and that is glucokinase, phosphofructokinase or PFK, and pyruvate kinase 1, 2, and 3 in red. During gluconeogenesis, 3 key enzymes are responsible for converting pyruvate back to glucose. That's PepsiK or phosphoenol pyruvate carboxykinase, fructose 1,6 biphosphatase, and glucose 6 phosphatase, with the first 2 highlighted in green. The next slides focus on the PepsiK as the third enzymes in this process, converting oxaloacetate to phosphoenol pyruvate, eventually to pyruvate. Now, Shown here are three irreversible enzymes involved in glycolysis on your left, along with the reaction they catalyze on the right. Once again, these are three enzymes that are essential for reversing glycolysis and transforming pyruvate back into glucose. Now, on ketone body metabolism or formation, now during starvation and diabetic ketoacidosis or DKA, the liver produces ketone bodies because oxaloacetate is depleted. This happened because oxaloacetate is diverted from the Krebs cycles to gluconeogenesis pathway. In DKA, the increase in gluconeogenesis is driven by low insulin level and elevated glucagon, which push the liver to produce more glucose without enough oxaloacetate to combine with acetyl-CoA. The Krebs cycle slows down, causing acetyl-CoA to accumulate and be redirected towards ketone body production. This excessive ketone production contributes to the metabolic disturbances seen in diabetic ketoacidosis or DKA. Now, the liver alone produces ketone bodies, functioning like a superhero that creates these molecules from fatty acids but doesn't use them itself. No? Since it lacks the enzymes to break them down, the liver releases ketone bodies into the bloodstream to supply other tissues, like the brain, the muscles, with an important alternative energy source when glucose is in short supply. That's why I often call the liver a superhero. Now, this is how extra hepatic cells metabolize ketone bodies. The ketone acetate or acetoacetic acid produced in the liver cannot be utilized there because the liver lacks the enzyme thioporase. In other tissues, acetoacetic acid is converted into acetyl-CoA, which then enters the Krebs cycle to produce energy. The extrahepatic tissues have sufficient oxaloacetate since they do not carry out gluconeogenesis. Now, the liver starts producing ketone bodies about 90 minutes after a high carbohydrate meal, but these ketones are rapidly used by organs like muscles and kidney. During prolonged starvation, ketone production becomes the primary energy source, allowing the brain to conserve glucose through excessive ketone levels can result in ketonemia. Now, during the post-absorptive phase, the kidney significantly contribute to glucose production by converting lactate into glucose through gluconeogenesis, especially in the renal cortex. While the liver is primarily the primary site, the kidney can provide up to 20 to 40%, or some textbook will estimate up to 50% endogenous glucose during fasting. This process helps maintain blood glucose level and is regulated by hormones like insulin, glucagon, as well as cortisol. Now, the kidney performs glycolysis mainly in the medulla, where low oxygen level favors anaerobic metabolism to generate energy. In contrast, the renal cortex carries out gluconeogenesis, converting substrate like lactate into glucose using key enzymes. This dual function allows the kidney to contribute to energy production and glucose balance, especially during fasting or metabolic stress. This slide shows that the medulla houses glycolytic enzymes while the cortex contains the gluconeogenic enzymes were previously discussed. So, with that ends the part 1. Part 2 will focus on the transport across cell membrane, movement of glucose into the bloodstream, and regulation of blood glucose level, as well as the mechanism by which cells take up glucose for energy use. So I thank you for listening.